I'm last year PhD student at IRB Barcelona in Patrick Saloy's group, the Structural Bioinformatics and Network Biology group. So today I'm going to present our latest manuscript that is currently under revision and is entitled Task-Adapted Biological Foundation Models Uncover Perturbation-Centric Representation. So the main question that we are trying to address here is that if we are able to adapt a general biological foundation model for being able to capture the effect that the perturbation exerts into a biological system. So when you perturb a biological system, for example, a cell with a perturbation like a compound or a genetic modification, you want to understand which is the effect that the perturbation is producing to the cell. And for this, one way of measuring it is through gene expression. So you can see which are the genes that go up or down after applying this perturbation, and this generates a molecular fingerprint of the response. These approaches have shown to be very useful for many downstream applications, for example, target discovery, informing mechanism of action, or even in drug discovery. So if you have a set of genes that you know that are dysregulated in a disease, maybe you can try to find a drug that reverts this pattern. So the objective of this project was to study these responses to perturbations using gene expression. And for doing this at the scale, we use the Lynx L1000 data from the Broad Institute. So this is the biggest public database of gene expression responses to perturbations. They measure more than 3 million gene expression profiles from thousands of different perturbations. So they can be compounds, but also silencing or overexpression or CRISP, many different types of perturbation. And they measure in hundreds of cell lines, and in different time points and different concentrations. So at the end, they end up with millions of profiles, 3 million of profiles. And for doing this, they developed this new platform. Well, in that moment, it was new, that is called L1000. And this platform, what it does especially is that it measures almost 1,000 genes, and then it infers the expression of the rest. So as you can imagine, this data is very powerful and very useful, but it also comes with some challenges. For example, the first challenge is this platform effect. So as it's a very specific platform in which it measures only 1,000 genes, then when you try to integrate with other data from other platforms or standard platforms like microarray or RNA-seq, sometimes it's difficult to integrate. Also, for some of the gene expression profiles, the quality is not as good as expected. So we saw low correlation between replicates and also batch effects. And also another characteristic of this data that we and others have described is that sometimes we see a strong cell effect. So this means that maybe two profiles are similar between them because of the cell line of origin, and maybe the perturbations that you have applied to this cell line are completely unrelated. So we were wondering if we can create a new approach in which we are able to extract which is the true effect that the perturbation is exerting and getting rid of all these confounders. So the data is what it is, but hopefully now we have better methods. So foundation models have become very popular in recent years because of the success in natural language processing and computer vision. So scientific community has started being interested in developing foundation models also for biology. So these foundation models are large models in which they are trained with large collection of data, and they show capabilities for which they have not been trained. So as I was saying, in recent years, a lot of foundation models have been developed for biology also, the majority of them using single-cell RNA-seq data. And in this case, the majority of them for training, they train in a self-supervised way in which they mask some of the expression of the genes, and then the objective of pre-training is to reconstruct the expression of the genes using the information from the other genes and also the general patterns of the data. So yeah, these models have shown to be useful for some tasks, for example, cell type annotation, but pre-trained doesn't guarantee that your model is going to perform good in your task of interest. So we were wondering that if we take a foundation model and we fine-tune it, so we adapt it, because one of the also characteristics of these foundation models is that they can be adapted or fine-tuning for your task of interest. So that is what we did in this project. So we decided to take one of the foundation models that is called single cell GPT, and then we adapt it for being able to extract which is the true effect of the perturbation without getting confused with the other confounders that we commented previously. So here you have a summary of our methodology. So as I was telling you, we use this linked cell 1000 data. And as I told you, each compound or each perturbation have been measured in different conditions. So this means that for the same perturbation, you can have thousands of different gene expression profiles. So you give this data to the pre-trained single-cell GPT model, and then for fine-tuning, what we put was an objective of classification, perturbation label prediction. So this means that the model has to learn from which perturbation label each gene expression comes from. And as I was telling you that for each perturbation, we have thousands of different profiles, the model has to learn which are the common things between all these profiles for being able to succeed at the task of predicting. So for doing this, the model, what it does is to transform this gene expression into a representation, into a latent representation that in principle capture this information for allowing the classification to perform good at this task. So this latent representation is the main objective of this project. So this is our perturbational embedding space, as we call, and this is what we are going to use for latest applications. So this classification was only a way of guiding the representation towards perturbation and not other confounders. So this is some of the downstream analysis that we design or that we can do with this representation. So for example, one of them, we can find similarities between drugs and infer with which is the mechanism of action of an annotated compound. Also, we can try to discover the drug target of an annotated compound again, and also, for example, we have this case that is contextualizing external experiments. So this means if a collaborator comes with new gene expression, maybe we can project it in our embedding space and tell them which are the perturbations that are more similar to the gene expression of interest. But for this, the first thing is to validate our perturbational space. So checking if the model if the fine-tune is producing representations that really capture the effect of the perturbation, so really organize the space according to the perturbation and not other characteristics. And here we are looking in 2 dimensions at 3 different perturbational spaces, so embedding spaces. So the first one is the pre-trained. So this means that we took the data and we give it to the model with the pre-trained weights without any kind of fine-tuning. And then we have 2 fine-tuned versions. So the first one was we fine-tune with the full links, and in the second one we fine-tune only with a high quality set of links according to their standards. So here we are coloring, so in the top row, we are coloring by the cell line of origin, and as you can see in the pre-trained, there are clear clusters, clear regions of the same cell type that are aggregated together, while this effect is diminished it's not as evident when you fine-tune. You can see some small cluster, but it's not as evident as it was when you pre-train. Then we color by the 8 compounds with more quantity of samples. And here you can see in the pre-train that the compound samples are spread in the embedding space, while when fine-tuned, you can see more aggregated regions according to the specific compound. Okay, but this was only 2D, and in 2D, distances can be distortion. So we decided to compute distances. So here again, we compare the pre-trained embeddings versus the two fine-tune. And here we have 3 sets of pairwise distances. So the first one were in purple, between embeddings from the same cell line. Then in blue, we have between embeddings of the same perturbation. And then in orange, we have unrelated profiles. Also, here, as you can see, in the top row, we have distances computed using embeddings. And then here we have distances computed using the raw gene expression. So yeah, as you can see here in purple, the distribution that these pairwise distances of the same cell line in both pre-trained and gene expression spaces, you see that they are shift toward the smaller distances. So this is the main factor guidance similarity. While the same perturbation and random things are mainly overlapping the distribution of distances. But this is not the case anymore when you look at the fine-tune. So here we can see a clear separation between the distribution of random embeddings versus the distribution of same perturbation. And also now we see that the cell line is not the main factor guiding similarity anymore. Now it's kind of in the middle. Okay, so we further quantified this using 2 complementary approaches. So first, we did this intra-perturbation label agreement. So this means that we compute for each specific perturbation, we compute within pairwise distances, and then we compare these within distances between the embeddings and the gene expression. And if the point is in the right, this means that the distances were smaller in the embedding. So as you can see for pre-train, only 26%, well, 27% of the cases significantly smaller than in gene expression, but this situation shifts completely when you look at the fine-tuned version. So here, the majority of points are significantly in the right part. So this means that the within distances are smaller in our embedding space than what it was in the original gene expression. Then we look at another approach using clustering. So here the idea is how unsupervised cluster explain the metadata. So in this case, cell line or perturbation. And we compare again the 3 embeddings versus 2 baselines, the gene expression and the PCA. So here you see that we fine-tune how well our clustering is explaining the cell line. It reduced significantly, while it increased significantly the how good it explains perturbation identity. So with all of this, we can claim that in general, globally, our embedding space seems to be organized at least better by perturbation identity than other type of characteristics, but we wanted also to see what happens in the local neighborhoods. So for this, we compute nearest neighbors, so which were the closest neighbors of each of the query embeddings, and then we check if in these neighborhoods we were able to see at least one neighbor with the same perturbation label than the query one. So for example, here we are comparing in the first neighbor, then top 5, top 10, and top 100. And again, we do with pre-train, fine-tune, and fine-tune with the high quality, and compare the 2 baseline, gene expression and PCA. So for pre-train, we cannot see difference with the 2 baselines, but then when we go to fine-tune, we can see a clear difference, a clear increase in performance for our embeddings. So for example, if we look at the top 1 neighbor in the high quality, we can see that for almost 70% of the cases, we are able to find that the closest neighbor share the same perturbation label than the query one. And if you go to 100, you see that at least one in almost all of the cases is sharing the same perturbation label. Okay, so now that we have validated that our embeddings are capturing somehow the effect of the perturbation. We wanted to distinguish if the model was only memorizing these perturbation labels or also capturing some relationships between the perturbations. So it's learning some biology of some perturbational biology. So for this, we evaluate how well the model was able to capture orthogonal information. For example, here in the case of compounds, in panel A, we benchmark how the embedding space was captured chemical similarity between compounds. So for this, we frame it as a classification exercise. So we define pairs of compounds that were chemically similar, and then as a scoring function, we use distances in the embedding space. Then we produce the ROC curve, and in blue, we have the results with our embeddings, and in red, we have the results with gene expression baseline. And as you can see, we increase our performance with an ROC of 0.81 versus what it was original for gene expression, an ROC of 0.64. Then we look at this local neighborhood consistency again, but in this time, what we were looking is that at least one of the 10 closest neighbors share the same mechanism of action that the query compound. And here we saw that almost in the high quality, almost for all of them, we were able to find in the top 10 that one of them were sharing the same, at least one of them was sharing the same mechanism of action label. And then in panel C, we did the same than for chemistry, but in this case, the pair were defining according if compounds share targets. And here again, we see an increase in performance for our embeddings in comparison to what it was the baseline gene expression. So we get an ROC of 0.74 versus an ROC of 0.58. As in our data set, we don't only have compounds, we also have, for example, silencing. We also explore some characteristic of the silencing perturbation. So here we see that we were able to capture relationships between different shRNAs that were targeting the same gene, and also that, for example, here in the local neighborhood, when you look at your closest neighbor, we were able to see that for 80% of the cases, it was sharing a gene ontology term. So it was related to your query one. We decided to explore a bit further this mechanism of action because we thought that maybe it's interesting for downstream applications. So here you have a 2D representation again, but in this case, we are coloring by the mechanism of action. So in pre-train, we don't see anything, any structure, we see a dispersion, but now with the fine-tune, we start seeing some regions that are specific of one mechanism of action. And also when we do this intra-label agreement, again, the majority of points are in the right part. So 80% for 80% of the cases, you significantly have smaller distances when you compare within the same mechanism of action. Okay, and finally, for comparing with other work and also for seeing if our embeddings can be used for predictive settings, so we are basing this exercise in this paper in which they do something similar, but instead of with a foundation model, they use an autoencoder. So they encode cellular response. So they also use links using an autoencoder, and then they compare different representations from the autoencoder to the raw gene expression in the benchmark of trying to classify perturbation class, manually perturbation annotated classes. So for doing this classification, they use both logistic regression and super vector machine. And as you can see from the results in the table, for all of the representation, they were not able to do better than gene expression. So in general, gene expression do a bit better than any of the representations that they tried. So we design the same exercise. We take their perturbation classes, and we evaluate again using logistic regression and support vector machine. So as you can see here for pre-train, we are performing worse than the baseline that was gene expression or PCA, the same that was happening here. But now with our fine-tuned embeddings, we are clearly outperforming the gene expression and the PCA. So what this means to us is that our embeddings can be also useful for in a predictive setting. Okay, so with all of this, we are happy with our representations. So they are capturing the effect of the perturbation, and they are also maintaining the known relationships between the perturbations. So we were wondering if we can use all these representations for extracting some knowledge. And the first exercise that we designed was mechanism of action inference. So from unannotated compounds, try to identify which is the mechanism from which they are doing their action. And for doing this, we are going to base on nearest neighbors. So here we have like an annotated sample of an annotated embedding of a compound. And then we are going to calculate their nearest neighbor, the top 50 nearest annotated neighbors. And then with this, we are going to transform the cosine distance into weights using softmax. And then we are going to sum all the different weights for the same mechanism of action. So at the end, we are going to obtain a mechanism of action distribution per sample. So this means that imagine that the red ones have the same mechanism of action. So we are going to sum their weights, and then we will have a probability for this mechanism of action. And the same for the blue or for the green one. But as every compound have more than one sample, we will have different probability distributions. So here what we are going to do is to average these probability distributions, and we are going to select the top one probability as the infer mechanism of action. This approach gives us two different metrics. So the first one is the confidence, that is the probability or the average probability of this mechanism of action. And then we will have the agreement. So for how many of these samples this mechanism of action was the top one. So with this approach, we were able to annotate more than 31,000 compounds that were unannotated in the links dataset, but we were wondering how good we are annotating these compounds as they are unannotated. And for this, we manually check the top 50. And for this, we were able to find evidence only for 36. And from the 36 that we were able to find some evidence, 33 of them agree with our predicted mechanism of action. So this manual validation was very encouraging, but we were wondering in a more systemic way, and also to see to what point we can believe in this confidence. So if there is a threshold in which we can say, okay, these predictions have a lot of probabilities for being correct, and these predictions maybe are noise, or maybe you should not believe it as much. So for this, we try to benchmark our confidence estimate. And for this, we design a benchmark in which we do it with the annotated samples. So we leave all the samples of one annotated compound out, and then we predict which is the mechanism of action using the same scheme. So for this, we were able to find that our performance was modest, but the good part was that the incorrect predictions always have low confidence scores. So as we can see here, this is in the x-axis we have the confidence score, and here the percentage of correct predictions. And as you can see, there is a clear positive correlation between having higher confidence score and having higher percentage of predictions. So if we play a bit with the threshold, we can see that, for example, if we put a threshold of 0.25, for more than 80% of the cases, you are going to give a correct prediction. So this is what we did at the end. And with this 0.25, we were able to produce almost 12,000 high-quality predictions for previously unannotated compounds. Okay, now we have the second application. So this is try to discover the compound target. So we were wondering if we link in our embedding space chemical and genetic perturbation, we can prioritize the target of the compound. For this, we were wondering if the perturbations, the genetic perturbations that are closer to a compound, if this means that this is the target or not of this compound. So again, we are based on distances, and again, each compound have several embeddings. So we compute distances from each sample to all the silencing embedding in the data set, and we will have a set of targets that are more close to every sample in our pool of our compound. So for this, instead of averaging the distances, what we did was a robustness-based ranking. So for example, in this case, HDAC is a gene that is in the top K of the 3 samples. So this means that it's 3 out of 3. So the frequency will be 1, and then in this case will be the first prioritized target. Then the same with other targets, for example, the EGFR. So in this case, maybe it's only in 2 of the samples of the compound in the prioritized list, and then it will be 2 out of 3, and it will be prioritized the second, and so forth. So with this, we wanted to evaluate how good our ranking was working, and we use a benchmark from the links, so a suggested pairs of compounds and genes that in principle should be correlated because these are the target of the compound. So these are 13 compounds for which we know the target according to this benchmark, and we saw in which position we were able to prioritize the correct target in this robustness-based ranking. So in blue, we have in which position we were able to prioritize, and then we have the random expectation. So as you can see, for some of them, we are able to do it very good, but then for all others, we do the same as random, or yeah, we prioritize in a point that doesn't make sense because maybe it's the first 2,000, so this is not informative. So we wanted to understand why for some of them we were able to perform correctly, but for others we don't have any signal. And for this, we manually inspect our list of prioritized genes, and we saw 3 different cases. So for example, there were some cases for which we don't have any information, so there is no clear target-related signal in this list of prioritized genes. But then there was one case in which, well, these 2 cases in which we were able to prioritize other target of the compound. So in this benchmark, we were looking for the aurora kinase B, but in our list, we found the aurora kinase A that was also described as a target. And same happened with roscotivin. So in this case, we were looking for CDK9, but we found CDK2 that it was also a target for this compound. There is another group of compounds that can happen another case. So for example, in this case, we have selutinib and this one, and in this case, we cannot find any target that is described as direct target for these compounds. But when we check at the list, we can see that there is many genes that are related to the mechanism of action of these compounds. So this means that you are capturing a coherent list of similarity between the genetic perturbations and the compound, but maybe you are not finding which is the direct target. So with all of this information, we thought that there is we can conclude two things. So the first one is that maybe it's difficult to do in this exercise to discover the direct target. Maybe we have to frame it as a mechanistic proximity. So yeah, similarity between the pathways that they affect, or yeah, they are plausible genes according to mechanistics, but maybe not the direct target. And also the second conclusion is that we need a way to distinguish between the predictions that make sense and the others that doesn't make sense, for example, these three. But for this, we need a biggest benchmark, because with 13 compounds, we cannot do a benchmark. We cannot design a way of scoring which predictions are good and which not. So for this, we went to literature and we found these two other benchmarks. So these are biggest collection of compound target relationships, and we applied the same framework. So in this case, we were able to correctly prioritize the direct target for more or less 10% in both cases, while the 90%, the rest of the data set, we were not able to prioritize the correct target. But when we were able to prioritize the correct target, we do it much better than random. So for us, what this means is that there is some signal, but we need to differentiate between a spurious correlation and the true signal. And yeah, one thing that we decided to explore for distinguishing between signal and maybe a spurious correlation was this transcriptional activity score. So this score was designed by Links, and this combines the signature reproducibility, accounting for quality of the signature. So this means if the correlation of the different replicates were coherent, and then the signature strength. So if genes, well, how strong were the changes in the gene expression relative to control? So here we have a 2D representation of our embedding space again, but now we are coloring by this score. And as you can see, it's not random. So we see some areas of high transcriptional activity score, and also we see some island of concentrated low transcriptional score embeddings. And also this is more importantly when you see in distances. So here we are comparing unrelated signatures, but we are separating the distribution between low TAS and high TAS. And sorry, TAS is the transcriptional activity score. So for the low TAS one, we see that it's shift towards smaller distances, towards more similar than in the case of high TAS. So this means that the embeddings that come from signatures that have low quality can be more similar between them, only because maybe they collapse in a place of the space for the embeddings from which you cannot extract a lot of information because maybe the raw data was very noisy. Okay, so with all this information, we decided to create a final score that accounts for the 4 different measurements. So the first one was the number of signatures, because as the ranking was based on robustness, we thought that maybe for the compounds for which we have more samples, this prioritization will be more robust. Then we have this task that I just explained you. Then score enrichment. So the enrichment means that you take the top prioritized genes, for example, the top 100, and then we check in, for example, KEGG on reactome of Go biological process, if there is some biological process that is enriched in this list of genes. So this will mean that maybe the prioritized genes are coherent between them. And then you have this score robustness, that is the average between the robustness score of these top 100 prioritized genes. So we aggregate all these 4 scores and we get a composite that, if you go to the previous benchmark, the one with these data sets, here you can see that it can significantly separate the ones that we perform good versus the ones that we were not able to find the target. So our composite score is able to separate these 2 distributions. And also, again, if we check how good, if we check how good our composite score correlates with the percentage of correct predictions, we can see again a positive correlation. So again, playing with the threshold, and taking into account a balance between coverage and performance, we decided a threshold of 0.35, which will guarantee us that more than 1/3 of the predictions will be correct. And with this, we are able to produce 1,500 lists of compound targets that, in principle, should be high confidence. Okay, and with this, the last application of our embedding space. So this is the contextualization, what I told you, like it's coming with new data and projected into our embedding space for exploring which are the perturbations that are more similar to your data of interest. And for this, we designed a benchmark exercise. So this benchmark exercise was based on links. So for the first one, we took the data of links and we held out 50 compounds. So all the samples of 50 compounds, we fine-tune again the model, and then we predict we project these 50 compounds that we have left out. And then we did the same, but with a specific cell line. So we left out all the different samples of a specific cell line, and then we project them. So here we have the results for the 50 compound out. So we fine-tune, and then we project these all the samples of these 50 compounds that we have left out. And here, chemical diversity, this is only for showing you that the compounds that we left out were chemically diverse. And here we have the results. So here for contextualizing, as there is no embedding of the same compound, we cannot check if the compound lands into a region that have the same perturbation label. So now for evaluating, we have to see if it lands into a region that share the mechanism of action. And we did it again with the neighborhood. So the top 1, top 5, or top 10, if at least they share one compound with the same mechanism of action. And here we can see a clear separation between what was random expectation, and for the top 10 neighbors, we see that for 50% of the cases, we were able to find at least one that shared the same mechanism of action. And then when we saw it in 2 dimensions, in bold, we have highlighted the points that we have projected, and then we have the colors of the different mechanism of actions. We can see that in general, they land into the region of their same mechanism of action. Then here we have the same, but with one cell out. So we took all the profiles of the A549 cell line, and we left all the profiles out. We fine-tune the single cell GPT model, and then we project these profiles that we have let out. So in this case, as the compound or the perturbations have been measured in many different cell lines, this means that now we can evaluate in the same perturbation label. So now we can see if the projection lands into an area in which all the perturbations are from the same label that the one that we are trying to contextualize. So here we see that our performance is modest, so we do better than a random, but the most that we can do is that when you look at the top 100 neighborhood, only for 25% of the cases, we are able to find at least one neighbor with the same perturbation label. And then when we go to the mechanism of action, we recover the performance. So again, we see a clear separation with random, and also for more than 50% of the cases, at least one of the neighborhood have the same mechanism of action that the one that we are projecting. So we were wondering if maybe we can improve this performance by providing some cell line context, because maybe this was difficult because the model have never seen anything about this cell line. So we designed 2 different ways of trying to improve the performance. The first one was only showing the controls of this cell line to the model and then projecting the perturbed profiles. And the second one was trying to adapt the fine-tune model. So fine-tune a model only without that cell line, and then adapt it with reconstruction loss using the controls of the cell line of interest. But as you can see here, there is no difference with the non-adapted version, so with this version that I show you here. So the results are completely the same, so modest performance, and yeah, we cannot see a significant increase in performance. So with this, we hypothesize that maybe this is because the number of samples that we are showing the model in comparison of that cell in comparison to all the other data for which the model is fine-tuning, maybe is very small, and maybe the model doesn't have enough information for learning from this specific cell line. And for seeing if this was the case, we went to our perturbational space, to the one that we fine-tuned with all the data, and here we check the Wasserstein distance versus the number of profiles that this cell contains. So this means that if the distance is small, it's because there is no, like, it's good integrated. So there is no difference between the distances or within the same cell line and outside the same cell line. And as you can see, there is a clear pattern that when the cell line have a lot of profiles, more than 10,000, yeah, they are very good integrated. So the model is able to really integrate the different cell lines and focus only on the perturbation effect. But then when the cell lines start having low numbers of samples, then we see that this integration is unstable. Okay, and yeah, the final thing that I want to explain today is this contextualization, but going outside of links. So imagine the real case in which a collaborator gives me data, and then I wanted to project it to see to which perturbation is similar. Okay, and for doing this, we went to Geo. We took an RNA-seq data set in which they measured the effect after 3 compounds, and these compounds were also in links. So we produced embeddings for these 3 compounds, and then we compare to the links embeddings of the same compound, so the training embeddings, but for the same compound. And as you can see here in the distribution, in blue are the distribution between the distances of the predicted or the external embeddings and versus the links versus random things. And as you can see, there is a clear separation in the distributions. And then when you look at the specific position in which you can find the correct compound, here we have 3 different cases. In the first one, we were able to find it at rank 1, so it was perfectly contextualized. We were able to find it the first position, then the 4th position, that was also very good, and for this case, we were able to contextualize it in the 60th position, which means that we do it pretty well because it's less than 1% of the data set. Okay, and now everything that I want to explain. So this led me to the conclusions. So we generated a perturbation-centric embedding space that capture biological effect more effectively than raw gene expression. Then that this embedding space recovers orthogonal information that was not explicitly trained on, that it enables some systematic knowledge extraction, for example, the mechanism of action annotation, and also this similarity landscape between compound and genes. And then we demonstrate some contextualization capacity. And with this, I would like to thank you all for your attention, also to my group, especially to my supervisor. And here, if you are interested, you have the manuscript, the code that we developed for this project, and also my email in case you have further questions. Thank you.
Thank you, Elena. Thank you very much indeed. My first question is about, of course, the biology is kind of hard sometimes to wrap your head around, but at least with a few details, it can be helpful. And for me, my first interest is about the embedding structure. One wonders, like, what do you think, like, the single embedding structure? regardless of the dimension that you chose, perhaps represent biologically, if at all it represents anything.
Sorry, I didn't get the question. Can you?
Okay, like what I'm saying is for the purposes of like visibility into the internal structure of the model, and like you have this embedding that is probably is a representation of biological hierarchy or entities. And I'm just wondering, like at a practical level, what does the embedding represent? Like, does it combine like the perturbations, the gene expressions? Like, uh, what does the embedding itself represent? How can we think
Okay,
about it?
okay, yeah. So now we're like in our mind what we think it represents. So what we are doing is transforming. So through this classification, we are transforming the gene expression into a latent representation that in principle is guide for being able to perform good in this classification problem of perturbation label prediction. So
Okay.
what we think that the representation is, yeah, is representing is the gene expression, but not the gene expression at all, like the raw gene expression is like extracting from the gene expression something that is useful for perform good in this perturbation label classification. But the model doesn't know anything about the perturbations, like we don't provide chemistry or we don't provide the labels of these perturbations. So the representation is only based on the gene expression that they produce, like, that is produced after perturbing a cell with this specific perturbation.
Okay, that's great. And maybe a follow-up question close to that. I was looking at that SCGPT, and in regards like to the final embedding structure, I'm wondering like how much comes from there is that data set, I think the links, yes,
Yes.
the links data set, like how much comes from one from the SCGPT, but also from the links data set, like that form that embedding that structure. Like what is the contribution of these two at the pre-training level and maybe at the fine-tuning level?
Yeah, so what we saw during the different exercises is that when you use the data as it is, so you take the data and you use the model as it is with the weights of pre-train, we see that the model is highlighting even more the effect of the cell line. So it's because I think it's the like the most evident thing that you can see when you see a gene expression profile. So there are many genes that change according to the cell line of origin. And then you have maybe some subtle changes that are because of the perturbation. That's why I think this changed a lot. Like here we can see when we show the model, the data, and we don't fine tune, we see that the model, what it does, the single cell GPT, the pre-trained weight, what it does is like extracting the cell line, because at the end, this model have been trained with 30 million single cell data points. So this means that this model have seen a lot of, like the objective, like I think the objective of this single cell GPT model was like cell type annotation, because it's trained with single cell data. So I
Right.
think that's why when you put the data as it is, then the model what produces an embedding that highlights the cell line of origin. But then when you fine tune, then you are guiding the model toward this perturbation information. So for the model to perform correctly in this classification, the model has to learn some way how to shift from only looking at the cell line to also getting information from the perturbation.
Thank you, Elena. There is a slide where you had some visual representations. I think about the main one was about cosine similarity, I think.
Here, okay.
Yes, I think this one, yes, the cosine distance. And my question is, why is cosine distance being the sort of like the right notion for similarity for these embeddings? Because well, I don't know, it could mean like the geometry of this perturbation maybe fits it, but one would think like why not Euclidean distance? Maybe so maybe you could say something about the choice of cosine similarity or cosine distance.
Yeah, so here there is two different answers. So the first one is because it was recommended by the authors of single cell GPT, and the second because, for example, in perturbations, we normally care about not the like we care about the directionalities, like that we are representing same direction, so that yeah, that the embedding go to the same direction because we think that this is what we care about, not the dimension itself, because maybe the when you measure gene expression, maybe yeah, you can have that some gene is super expressed, so you will have a very big number, but still is also overexpressed in other, but maybe in a lower number. And this many times doesn't mean that it's because it's more expressed or less. Also can be because of the way in which you are measuring the gene expression. So normally you care more about the directionality, that you are going into the same direction more than the actual magnitude of the change.
Okay. I think my there would be maybe last or second last question is about we observe that I think somewhere you showed that like compounds with the same mechanism of action with the same or MOA they tend to become close in the embedding space.
Yes.
And my question would be at that point what can we say about the model learning? Like, what does the model actually learning in that instance or in that situation for these similar compounds and their embedding space coming close?
I think that the model what is learning is so at this learning, which is the effect of the perturbation, like is learning from the gene expression, which are the main specific genes, or yeah, which is the main specific part of the gene expression that it has to check for for understanding which is the effect of the perturbation. At the end, if two compounds have the same mechanism of action, this means that they will affect similar pathways or similar part of the gene expression of the cell. So at the end, this for us is like a validation that our model is actually learning what it should. It's learning like biology because it's able to put together things that should be together, because in principle they are messing with the same part of the biology of the cell.
Okay, I think this is an interesting discussion. I don't see questions from the chat. And members, if anyone has a question, please unmute yourself. If no one, I want to say uh, goodbye to Elena, but before that, I want to really thank her for saving up time and really talk to us about this work. Thank you very much, Elena.
Thank you.
I know the PhD is really a great journey. So thank you very much, and please deliver our thanks to the professor. We
Okay,
are really grateful. So thank you very much.
thank you so much for the
Uh,
invitation and the discussion.
all
Bye.
right. Bye bye and goodbye to everyone. See you in our subsequent sessions in the coming weeks.
Bye.
Bye bye.